Okay, thanks a lot, and thanks for giving me the opportunity to speak and share what we propose as a unified approach to causal mediation analysis for, say, the typical. Okay, sorry, yeah, that was what I, yeah. Okay. Is that better now?
That's good,
Yes.
Go ahead.
Okay, great. Okay, yeah. Thanks for giving me the opportunity to speak and share what we propose as a unified approach to causal mediation analysis for, say, the typical phase 3 trial in the pharmaceutical industry. The methods have been developed in collaboration with Ghent University, first and foremost, Professor Stijn van Laand, and also my good colleague Martin Linder from Novo Nordisk. And as described in 2 papers, one with a focus on time to event income, and the more recent paper with a focus on continuous or a binary outcome measured at a landmark visit. The methods in the 2 papers are conceptually similar, although slightly more complex in the case of time to event outcome. The methods can take all relevant longitudinally measured, adjust for sources of measured confounding. And actually, this is not too complicated, in fact, because the estimation method uses just standard regression models in a recursive manner. But more importantly, the reason for us doing this work is because in the past years, we have experienced an increasing interest from clinicians and regulators in questions related to mode of action, as illustrated by the title of the paper on the right bottom. And here the compound in question is tirzepatide. It's a compound that shares mode of actions with the so-called GLP-1 receptor agonists. And let me explain a little bit more on this specific clinical question of interest. So the well-established effects of these GLP-1 receptor agonists are via 2 main pathways. So one is the effect on glucose lowering via insulin secretion and glucagon inhibition. And secondly, the effect on weight loss via feelings of satiety. There is furthermore also a general understanding that substantial weight loss will imply glucose lowering via increased insulin sensitivity. And so the clinical question of interest here is to assess how much of the total glucose lowering is due to the weight loss. And now, let me try very, very quickly and briefly to describe sort of the concepts of the methods. First, to the estimands. And for simplicity, we will illustrate the estimands that we refer to as path-specific effects in a setting where there are only 2 post-baseline visits, post-randomization and randomization to either GLP-1 receptor agonist or placebo. And so here, Y1 and Y2, this denotes measures of the outcome. So in the example, it's blood glucose measured in terms of HbA1c. M1 and M2 are measures of the candidate mediator. In the example, it's change in percentage body weight. And L1 and L2 here denotes potential post-baseline confounders. In the example, it could be initiation of additional diabetes medication. C0 denotes relevant post-baseline covariates. Then in the corresponding causal diagram on the right, the causal pathways from A to the final outcome of interest Y2 that immediately or initially acts on one of the 2 measures of the mediator. These pathways are counted as part of the intermediated effect, and they are here marked in blue. Correspondingly, all other pathways are counted as part of the direct effect. Note that effects directly impacting a post-baseline confounder and then in turn the mediator is here counted as a direct effect. Now this is causal inference, so of course the estimands illustrated here are defined more rigorously in terms of of potential outcomes, and the key potential outcomes in mediation analysis are the so-called cross-world potential outcomes. And here an example denoted as Y210 would be the outcome if a subject received active treatment, but where the mediator values M1 and M2 were set as if the subjects were receiving placebo, however, without changing Y1, L1, and L2. Lastly, let me briefly explain about the assumptions for identification and the estimation. So the assumptions for identification are the usual consistency or positivity assumptions, and that the causal diagram represents a non-parametric structural equation models with independent errors. These assumptions are made in many causal inference applications. Except for that, then the main assumption, and it's the assumption that we also try to discuss with our clinicians, is that there are no unmeasured common causes of mediators and outcomes, and mediators and confounders, respectively. However, note with the green arrows here that some unmeasured common causes are in fact allowed, such as between confounders and outcomes, or by the repeated measures of the mediator. Under these assumptions, we derive a G-formula-like expression for the cross-world's potential outcomes, and we propose recursive estimation in this expression using simple standard regression models. And finally, I would like to mention that we also develop SAS macrocode, which is available with the papers that I have given a link to in this presentation here. And with this, I'll end this extremely brief introduction. But anyway, I hope you could follow some of the concepts here. And I'll just like to say thanks for your attention.